unusual day, one that comes only every 4 years. And it's really fitting that on an unusual day, we're also hosting an unusual IMED lecture. Many of us in the room, many biomedical scientists, really strive to make discoveries that we can take from the bench to the bedside. And some seminal discoveries have been made here at UCLA, both in devices and in scientific bench-to-bedside translational research. But many of us haven't been that lucky to be able to discover something that we can actually take to patient care. And today we're going to hear one of those unusual stories, or rare stories, and one that's received a lot of press over the past month. So we're honored today to have both the scientific side and the clinical side of a bench-to-bedside translational work. In case anybody hasn't seen it, Lanza just published on February 25th a paper that's first authored by Dr. Steve Schwartz and last authored by Dr. Bob Lanza, both of which are talking to us today. And the title of this is Embryonic Stem Cell Trials for Macular Degeneration, a Preliminary Report. This is the first description of human embryonic stem cell-derived cells transplanted into human patients. Dr. Lanza is the Chief Scientific Officer at Advanced Cell Technology and an adjunct professor at the Institute for Regenerative Medicine at Wake Forest. However, his first publication at age 18 was a single-authored publication in Nature, where he started his genetic quest, where he was studying chickens. Most of us did not have a first-authored, single-authored publication in Nature at age 18. He subsequently went to the University of Pennsylvania, where he received both his BA and MD degrees. He had a Fulbright, among many other awards in his life. He's worked in the laboratories of such luminaries such as Jonas Salk, Gerald Edelman, Rodney Porter. He's worked with B.F. Skinner and has several publications with him, and also heart transplant pioneer Christian Barnard. He's been in the company of many great people over the years. He has hundreds of publications and inventions. He's got 24 books, among them Essentials of Stem Cell Biology and Principles of Tissue Engineering. Last year he won the NIH Director's Award for translating basic science discoveries into new and better treatments. The 2010 Movers and Shakers, Who Will Shape Biotech over the next 20 years, along with luminary Craig Venter and President Barack Obama. Keeps really great company. Equally as illustrative, Stephen Schwartz is the Amundsen Professor of Ophthalmology. He's been the chief of vitreoretinal surgery here at UCLA for a decade. He unfortunately did his bachelor's at Cal, and then went to our competitor, USC, for medical school. But then he actually saw the light and came here as a resident to the Jules Stein Eye Institute of UCLA. And then he pursued additional fellowship training in retina at the prestigious Murfield Eye Hospital in London. During his tenure here at UCLA, he's been both an active clinician and a clinician investigator. He's participated or led or designed multiple early-stage clinical trials for novel retinal therapies, both in the device, drugs, and translational realms. He was pivotally involved in anti-VEGF therapy and performed both phase 1 and phase 2 trials here at UCLA. In addition to those activities, he's mentored numerous residents and fellows, and he's been on the scientific advisory board of many companies. So with that brief introduction, I'm going to turn this over to two really luminaries who will teach us much about the first in-human clinical trial of human stem cells. Dr. Lanza.
Okay. Thank you very much, Lynn, for that very kind introduction, and it's a great honor for me to be here today. And as you can see, I plan to talk about pluripotent stem cells and what's next for the clinic. As you may know, there's two major hurdles in regenerative medicine, namely the shortage of tissue and the problem of immune rejection. So this graph here shows you that there are a lot more people on the waiting list than there are currently available donors. But in addition to the shortage of whole organs, there are literally dozens and dozens of human diseases caused by tissue loss or dysfunction, including diabetes, for instance, caused by a loss of the insulin-producing cells in the pancreas, or the loss of the RPE cells in many eye disorders. Now, with the advent of stem cells, we now have before us an opportunity to solve both of these major problems. And once we have an unlimited source of source material, hopefully we will then be able to use tissue engineering to reconstitute these cells into more complex tissues and structures. In order to create immune-compatible cells, there are two major approaches. There's one known as SAMT, which is essentially therapeutic cloning. And in this approach, you basically take a cell from the patient you would want to create the stem cells from, place it next to an empty egg, and then send an electrical charge through the unit. Two cells fuse, and the nucleus of the cell you want to clone gets dumped into the empty egg. You forward into thinking it's been fertilized, it starts to divide, and you create embryonic stem cells. The second possible approach is through cell reprogramming. And in this approach, what you do is you upregulate certain transcription factors and actually bring that cell back to a pluripotent cell to a state they're called induced pluripotent stem cells, or IPS cells for short. Now, there are both positives and negatives to both of these approaches. For SCNT, one of the pros, of course, is that you could theoretically create an unlimited source of immune-compatible tissue. On the downside is it requires human eggs, which are very scarce, as well as it requires the generation of embryos, which has ethical concerns. And finally, perhaps most importantly, is that we don't quite have that technology working. More recently, the IPS cell technology has been developed, and I'll touch base on that in a minute. But basically, whether it's through SCNT or IPS cell technology, this is an experiment I carried out quite a few years ago just to show you the power of this technology. So these are some old cows that we found that were about the equivalent of humans that are 70 or 80 years old. We took a skin cell from the ear and created cloned hematopoietic stem cells. And then we injected those back into the vein of the nuclear donor animal. And because they were so youthful, we didn't need to use any myeloablation. The cells, through competitive repopulation, were able to set up home in these animals very nicely. And because they were cloned cells, we had to put a marker in, a neomycin cassette, so that we could distinguish the cloned cells from self. And what you see here is the appearance of the cells over the first 12 weeks in the blood of these animals. And amazingly, a half a year, 6 months later, when you took a sample of peripheral blood and plated it out, you would actually see these giant white blood cell colonies. These are from the colony-forming cells. In 60% of the cases, you only see these giant white blood cell colonies in very youthful animals. So the goal here wasn't just to give an old cow a new immune system. It turns out there are over 80 human autoimmune diseases, lupus, multiple sclerosis, rheumatoid arthritis, that these type of strategies could potentially help. This is another study that we carried out with Piero Anna Versa, where we actually were studying a myocardial infarct model. So this is a cross-section of the heart. This is the healthy tissue. This is the infarcted tissue. And we injected cloned CD34 cells at the margin of the infarct. And within 1 month, this was LAC-Z positive cloned cells, about 40% of the damaged tissue was restored, and there was a very robust vasculature as well. So that shows you what patient-specific cells can potentially do. And the more recent approach, again, is deriving these IPS cells. So again, not only can you make unlimited sources of patient-specific cells, but unlike SCNT, no eggs are required, no embryos are generated. You take a skin cell and turn it into a pluripotent stem cell. The disadvantages, unfortunately, is that the two major approaches that have been used, the Thompson and Yamanaka method, require the use of lenti and retroviruses, which unfortunately, we published a paper showing for the first time that they actually were able to, the cells that you turn them into had quite severe problems, apoptosis, very severe limitations in their growth and expansion capacity, as well as they displayed early senescence. And this slide here just gives you an idea of what I'm talking about. We try to create red blood cells from both embryonic stem cells and IPS cells, both through the retrovirus and lentivirus method. And what you have to do is you create these blast colonies, and you go through various cycles of expansion to get your erythroid cells. And what we found is that, as we've seen in the past, is that we get very nice large BC colonies with the embryonic stem cells. By contrast with the IPS cells, they're very small, they don't expand very much. So after 3 cycles of differentiation and expansion is literally over 1,000-fold difference in expansion capacity between the 2 types. So this is very severely limiting for clinical translation. Fortunately, with some of the newer technologies, we've now, we think, solved most of those problems. So here's some work that published with Dr. Kim at Harvard, where we actually generated these IPS cells by direct delivery of reprogramming proteins. And there are new methods, other methods that do not require viruses as well. And so those methods are up and coming, but until that happens, for the moment, while we're still dealing with immune rejection, the most likely first translations are going to be in immune-privileged sites, such as the brain and the eye. The eye is especially attractive because obviously there are many major visual disorders such as AMD that cost the U.S. healthcare system over $50 billion a year. Unfortunately, this slide, so there are many different cell types that could potentially help regenerate the eye. One is the retinal pigment epithelium. Another one is the angioblasts, which I'll talk to you briefly about. Another one more recently we've been studying these retinal neuroprogenitor cells. We're also doing some work with corneal endothelium and also with MSC mesenchymal stem cells. And I'll touch briefly on each of these. So first I'd like to talk to you about treatment for retinal degenerative diseases. We recently initiated 2 clinical studies involving the use of RPE, or retinal pigment epithelium, to treat age-related macular degeneration, as well as Stargardt's disease, which is the leading cause of juvenile blindness. And AMD alone affects over 30 million people worldwide. It's the leading cause of blindness in the developed world. The degeneration of the RPE plays a critical role in the development of AMD, Stargardt's, as well as literally over half a dozen other retinal degenerative diseases. And we've also carried out animal studies showing that the transplantation of these RPE cells can rescue both photoreceptors and attenuate the loss of visual function in these animals. And so for that reason, we believe that stem cells could be a very potentially interesting source of cells for RPE transplantation. So this is a cartoon of the eye. The light comes in through the cornea, and it hits the photoreceptors, the cones and the rods that we see with, and underneath that are the retinal pigment epithelial cells, which are responsible for maintaining the health of the photoreceptors, as well as many other functions, ion transport, etc. And also importantly, they phagocytose the shed photoreceptor segments. And there are many advantages to using pluripotent stem cell-derived tissues for regenerative medicine. You could create virtually an unlimited supply of cells. These cells are immortal, they grow forever. You can drive these under GMP conditions, under pathogen-free conditions. They can be produced with minimal batch-to-match variation, and they can be thoroughly characterized to ensure optimal performance. And we've now studied literally 2 or 3 dozen of these embryonic stem cell lines, and they all reproducibly generate RPE that can be passage characterized and expanded. And this just shows you cultures of differentiating embryonic stem cells. So virtually all the cultures will eventually spontaneously start to develop these little freckles in the Petri dishes. This is what they look like under harder magnification, and you can mechanically dissociate those cells. And then once you dissociate them, they start to lose their pigmented cobblestone morphology, and they continue to grow until they reach confluence, at which time they reestablish their RPE phenotype. And they can go through this differentiation, transdifferentiation many, many times, so that you can literally create tens of millions of these cells. In fact, 1 6-well plate is enough, can create enough cells to treat 50 to 100 patients. We have extensively characterized these cells. We can show very clearly that there's a downregulation of all the markers of pluripotency, and also an upregulation of all the RPE-specific markers. We've also develop some potency assays. So for instance,